Yeah, it is. I would suggest that it totally depends on, like, you first decide what course of career you want, because if you want to stay in research, then these kind of extra courses and even taking agriculture is a good option, because then you can join different ICAR institutes as scientist positions, and it will become more easier for you to join because you will have techniques which they require. So if that, like, decide first that what you want to do after your masters, whether you want, because if you want to be in science, that means you have to go for PhD. So are you ready to invest that much of time in PhD? And if yes, then you can decide these things. Yeah, see, biotechnology is very diverse field, so that will help you in understanding a lot of concepts which your core subjects might not help, and it will be helpful if you are taking research as your career option. But like, according to some people, it might be a little difficult if you want to pursue in like teaching, because there are chances that vacancies are more in core subjects, and also some institutes don't consider MSc biotechnology as eligible degree for teaching botany as subject. So there you might, I mean, I myself have done MSc biotechnology, but I have not yet applied for assistant professor, so I'm not sure. But like, from what I can see, people are facing this issue. I am not a botany student. Yeah, not yet. I mean, I'm in my final year, but yeah. Okay. There is no point, because you will just be wasting your time. Don't go for that, because you've already done bachelor's. It would have been better if you would have done BTech. You could have directly joined for PhD, because that facility is there now. But if you've already done a graduation, don't go for another graduation. Rather join some integrated courses. It will be easier for you. Some MS PhD courses. Yeah, sure, sure. Yeah. So we'll just start with the session. Good evening, everyone, and welcome to week 9 of live interaction session for basics of crop breeding and plant biotechnology. And I am Prashasti Pandey, PhD scholar and PMRF fellow from Department of Botany, Banaras Hindu University. And if you have already attended previous live sessions, you know that in these sessions we'll check some questions from previous year's assignment, and I'll give you some time. You can try solving them. If you know the answer, you can write it in the chat box, or you can speak it out loud. And we hope that these will help you in solving this year's assignment for this course. Okay, so let's proceed. So the first question is on your screen. Choose the correct statement regarding DNA polymerase 1. Options are: it synthesizes new DNA strand by adding nucleotides to a primer. It's involved in the proofreading activity. Its nucleus activity degrades the existing strand as it proceeds, and it lacks nucleus activity. It is an MSQ type question, so multiple answers will be correct to this. Option A. Okay. Okay. A and B. Anyone else? Okay, so let's understand this question. This question is talking about DNA polymerase 1, right? So let's understand first that what is DNA polymerase 1. It is basically a prokaryotic enzyme, okay, and it is involved in DNA replication and its repair. So, in these kind of terms, you can understand the function of that particular thing by breaking down the words. So, like if you're talking about DNA polymerase 1, so it has something to do with DNA. And because polymerase word is used, polymerase everywhere when we are talking about enzyme is usually used when we are trying to produce some new DNA. Okay, it can be from a DNA strand or it can be from a RNA strand. So the activity of DNA polymerase 1 will be in DNA replication or in repair, because both replication and repair will require uh, preparation of DNA, even in case of repair. So if this is one strand and there is some damage to the DNA in this particular place, then adding the particular complementary base pair in this site will also require addition of new bases. So that is also a part of polymerizing DNA, or basically synthesizing DNA. Even though it is a part of DNA, but it is still synthesizing the DNA. And replication involves complete forming of new DNA strand from the parent strand. Okay, so then again it involves making a new DNA strand. So both these activities comes under the function of DNA polymerase 1, and this is DNA polymerase. Basically, this is the function of DNA polymerase. DNA polymerase 1 specifically is a prokaryotic enzyme which is more like specially present in certain bacteria like E. coli. Now it has three important functions. Okay, first function is 5' to 3' polymerase activity. Now, if we are aware of the basic concept of DNA, it has two strands. Okay, one strand is 5' to 3', and the other strand is 3' to 5'. One is called as leading strand, which is 5' to 3', and the other one is called as lagging strand. Okay, now when the first function says that it has 5' to 3' polymerase activity, that means it is only able to synthesize in this direction. It cannot synthesize a new DNA strand in the opposite direction. So if it is synthesizing from this direction, that means the template will be 3' to 5', and the synthesis will be in this direction. That means the new strand that is being formed will be equal to the 5' to 3' direction, and it will make a straight new strand with the complementary base pair of the other strand. Okay, so 5' to 3' basically it adds nucleotides to synthesize a new DNA strand from using primers. Okay, second activity is 3' to 5' exonuclease activity. So now this exonuclease activity is also called as proofreading activity. So what happens is, while this particular polymerase is synthesizing a new strand in 5' to 3' direction, it also has, because it's an enzyme, right? So it will have certain uh, functionally active domains which can perform certain functions. So if you look here in this figure of DNA polymerase 1, it has a domain for synthesis, it has a domain for proofreading, and it has a domain for primer remover. Okay, so if this particular domain is synthesizing in 5' to 3' direction, it can also simultaneously proofread the DNA in the opposite direction. So while the synthesis is happening in this direction, this particular enzyme also keeps on checking whether the added base pairs are correct and they are complementary to the strand it is making a new strand from. Okay, so this activity is called as proofreading activity. And if it finds that there is some base pair which is not the required one. So uh, suppose the template strand says 80 GC 80. Okay, so the correct synthesis of this opposite strand should be TACGTA. Right, now suppose for some reason uh, instead of this a T is added. So while it is synthesizing here, once it reads C, the proofreading part will check whether this base pair is correct or not. And if it finds that the two base pairs are not correct, it will remove this base pair. Okay, so this particular activity is called as proofreading activity, which is in opposite direction, that is 3 dash to 5 dash. And the third function is 5 dash to 3 dash exonuclease activity. So the third activity, what will happen is when the new DNA strand is synthesized from the old DNA strand, there is a small primer sequence which is added from here, and then the DNA polymerase will bind here and the synthesis will start. Right? So the function of 5 dash to 3 dash exonuclease activity is to remove this particular primer. Okay, this will remove this RNA primer or any damaged DNA which is ahead of the growing strand, and it will replace it with correct DNA. If the primer here that is used is of RNA, then it cannot be the complementary through DNA, right? Because RNA will have uracil. Okay, so it cannot be complementary to the DNA. So for it to be correct, it will remove this primer and instead replace it with the DNA bases. Okay, so that is how it will correct. And if there are some other bases which was removed because of the proofreading activity, this particular polymerase will also add those base pairs. So these are the three main important functions of DNA polymerase. One is 5 dash to 3 dash polymerase activity. The other one is 3 dash to 5 dash proofreading activity. And the third one is primer removal or removal of damaged DNA and adding of the new base pairs. Right? So again, talking about the role of this, the important function is that it can remove primer from Okazaki fragments. Okazaki fragments are formed from the opposite strand where um, the strand synthesis is happening in opposite direction. Okay, that is on the lagging strand, and then filling those with the gaps, because in case of lagging strand, the synthesis will happen in small fragments. Okay, synthesis will happen like this. So there are primers attached everywhere, and those primers are RNA primers. So these have to be removed. So uh, basically, this polymerase will remove this, and it will add DNA bases instead of this primer. Okay. So, in simple words, DNA polymerase 1 is a bacterial enzyme that will remove RNA primers. It will fill gaps with DNA and proofreads the new strand. Okay. And also, it was the first DNA polymerase to be discovered. And it is not the main enzyme for bulk DNA replication. Uh, if there is a bulk DNA replication happening inside bacteria, mostly DNA polymerase 3 is used, but it will not have proofreading activity. So for that, DNA polymerase 3 is used. Okay. So now, coming back to our question about the four statements. The first was it synthesizes new DNA strands by adding nucleotides to the primer. So this statement is true because yes, it can extend from 5 dash to 3 dash direction by adding nucleotides to existing 3 dash end. Second statement was it's involved in proofreading activity. This statement is also true because it has 3 dash to 5 dash exonuclease activity, which will remove the wrongly added nucleotides. Third statement was its nuclease activity degrades the existing strands as it proceeds. Yes. So in case of Okazaki fragments on the leading strand, it will degrade that RNA primers and the faulty nucleotides ahead of it, and it will replace it with the new DNA bases. Okay, so this statement is also correct. And the fourth one was lacks nuclease activity. This statement is wrong because DNA polymerase has both direction exonuclease activity, 5 dash to 3 dash, as well as 3 dash to 5 dash. So the fourth statement is wrong. So according to this, the correct answer to this question, because it was the MSQ, all A, B, and C options are correct. Okay, only D option is wrong. So accepted answers will be your A, B, and C. Okay, so I hope this concept was clear. Let's move on to our next question. Which of the following enzymes is involved in the removal of 5 terminal phosphate group? The options are alkaline phosphatase, polynucleotide kinase, reverse transcriptase, and terminal deoxynucleotidyl transferase. Okay, we have one answer is option A. If anyone else knows the answer. Okay, so let's talk about this question. So let's first understand all the four terms, four options which was there in the question in very short way. So the first was alkaline phosphatase. Okay. So function of alkaline. So now again, I told you that if you understand the meaning of the terms, it will become very easier to understand its function, right? So phosphatase term, phosphatase is usually used when a phosphate group is being removed. Okay, phosphate phosphate should be removed in case of phosphatase. Okay, and in case of kinase, wherever kinase is word is used, in this case, phosphate here because it is added with phosphate, right? So whenever uh, kinase is used, in that case, the phosphate term will be added. Okay. So this is the meaning of these two terms, phosphatase and kinase. So now, in case of alkaline phosphatase, basically it will remove the 5' phosphate group from DNA or from RNA or even nucleotides. Okay. So the main, so when is it useful? When we are having a vector. Okay. And now we are digesting that vector and it is cut from some, suppose RE enzyme or some blunt enzyme. And now it is open. Now we need to use this for cloning and we need to add our insert here, right? But if there is an open phosphate group on this side and there is an open hydroxyl group on this side, then in that case, sometimes there are chances that this vector will self ligate itself without this insert. Okay, so when this insert will not be added, the vector will get self ligated and it will also get transformed. Okay, it will get transformed in E. coli. It will give you the similar results when you plate it on a selection marker plate. It will get selected. And then later, when you confirm it either by running a PCR with the plasmid or using restriction digestion, then you will get to know that this insert was not added in the plasmid because the vector got self ligated. So, in such cases, to prevent this entire self ligation thing, we use alkaline phosphatase. So what it will do is it will remove the phosphate group from here. So now phosphate group is removed. That means there are no chances for the vector to self-ligate, and only an insert can be ligated in that place, because that will have the that will have that end. So this usually is used when we are keeping digested vector for a little longer time, or when there are high chances of the vector being self ligated, or there is some enzyme used where there are compatible ends produced in both the sides during digestion. So in those cases, alkaline phosphatase is used, and it will prevent vector self ligation during cloning by dephosphorylating the DNA ends. Okay. The second term was polynucleotide kinase. So I told you the function, that in this case, the phosphate group will be added to the 5' OH end of the DNA or the RNA. Okay, so now it will label it. The labeling will happen at the 5' end of all the nucleic acids. Okay, and the phosphorylation will end before the ligation. Now the common example is T4 polynucleotide kinase, which can do the function of adding a phosphate group to the 5' OH end of the DNA strand. Third term was reverse transcriptase. Now again, if you take this word, basically transcription means conversion of our DNA to RNA. Right? Now, if we do the reverse process, that is, if we go from RNA to DNA, that process is called as reverse transcription. The only difference is that now it won't be cRNA, it will become cDNA, right? It will become complementary DNA. Now, what is the difference between DNA and cDNA? Okay, so the major difference is that because this cDNA is formed from RNA, our while forming RNA, the so we know that in DNA there are two types of structures, introns and exons, and only exons get transcribed to form the RNA. That means if we are going back from RNA to DNA, in that case, there will be no introns present in that sequence. So, the DNA sequences which lack the intron and the sequences are complementary to the RNA strand is called as the cDNA or the complementary DNA. Right? So, reverse transcriptase is the enzyme which helps us do this procedure of going from RNA to forming cDNA. Okay, so here it's written exactly the same, that the main function of reverse transcriptase is forming of cDNA synthesis, and this cDNA will can later be used for performing RT-PCRs and studying of various gene expressions from mRNA, because the DNA content in all the conditions will always be same, but the RNA forming from that DNA will differ on the situations or the conditions in which the cell is. In certain cases, there might be expression of the gene where the gene is getting converted to RNA, but there might be normal conditions where this is not happening. So, the level of RNAs which can be studied from forming the complementary DNA, because more the RNA, more the amount of complementary DNA will be formed. So, we can predict whether the gene expression has changed or it has varied, either reduced or increased in certain conditions, by studying the complementary DNA. And the complementary DNA will be formed by the use of the enzyme called as reverse transcriptase. Okay. And the fourth word was terminal deoxynucleotide transferase, which is also called as TDT. Now, the function of TDT is to add deoxynucleotides to the 3' end of the DNA without needing a template. Okay, so it will add deoxynucleotides, and there is no template required for that. Now, this the main use of using TDT is to add a homopolymer tail to the DNA. And there are certain assays like tunnel assay for detecting DNA breaks, and some nuclear biology techniques which require labeling and certain kind of things where deoxynucleotides, some sequencing also requires addition of deoxynucleotides. So that can be done by PDT. Okay, so these are the meaning of four functions. Four functions of the four terms that was given in the question. Now coming back to our question, that removal of 5' terminal phosphate group from DNA or RNA is done by alkaline phosphatase. Okay, this is commonly used in, we talked about it, that it is used in dephosphorylation of the vector DNA in cloning and to prevent self ligation of the vector. Why the other options were incorrect? Because we have talked about their functions. Polynucleotide kinase will add phosphate instead of removing it. Reverse transcriptase will synthesize the cDNA from RNA, and TDT will add nucleotide to the 3' end of the DNA. So the correct option to this question is option A, alkaline phosphatase. Okay, I hope the concept of this question was clear. Let's move on to next question. Reverse transcriptase enzyme is RNA-dependent RNA polymerase, DNA-dependent RNA polymerase, RNA-dependent DNA polymerase, and DNA-dependent DNA polymerase. Okay, so let's talk about this question. So let's talk about the four terms again, which was given in the option. The first term was RNA dependent RNA polymerase. Okay, so in these terms, it is very like you can understand from the question only. RNA dependent RNA polymerase means that the template will be RNA and the strand that is produced here will also be of RNA. Okay, so this means that the polymerase will work on the RNA template and it will produce another RNA template. Okay, so this is what RNA dependent RNA polymerase does. It will use RNA as the template and it will make the RNA. So now it is where it is found. So it will be basically found in those organisms which have their genetic material as RNA. And that is why during replication, they require again RNA to be produced. So this happens mostly in case of RNA viruses. Okay, so this will happen mostly in case of RNA viruses and or in cases where replication is needed for viral RNA genomes. Like okay, so there is an example like replication of some plant viruses or some RNA viruses will require RNA dependent RNA polymerase. So you can understand this from this figure that uh, uh, in case of if the template is a wireless genome of single stranded RNA and it will get translated to form the viral protein. But now, if it requires multiple copies, in that case, RNA dependent RNA polymerase will basically use this single stranded RNA as a template to form the complementary single stranded RNA template. And again, this RNA dependent RNA polymerase can form multiple copies of this particular template. Okay, so this is the function of RNA dependent RNA polymerase. The second term was DNA dependent RNA polymerase. So in this case, this enzyme, so again from this term, DNA dependent RNA polymerase, that means there will be one strand, the template strand will be DNA, okay, and the strand that is being produced will be of RNA, okay. So this enzyme will use DNA as the template and it will make RNA. So now the function, this is basically RNA polymerase function, which will be used in transcription because your DNA in the transcription process, DNA is used as a template to form the complementary RNA. Okay. And it will have role in forming mRNAs, rRNAs, tRNAs from DNA. rRNA is basically ribosomal RNA, tRNA is transfer RNA. Okay, so in this figure also you can see the same thing. In so here it is given in terminology for if the template is a single stranded RNA, then this RNA with the help of RNA dependent DNA polymerase get converted into single stranded DNA. And from this DNA dependent DNA polymerase will convert to double stranded DNA. And now it can get converted to RNA also using DNA dependent RNA polymerase, or it can produce multiple strands of this particular thing. Okay. So this figure is utilizing three types of polymerases. But I hope this function of DNA dependent RNA polymerase is clear. Third is RNA dependent DNA polymerase. Now it is again that the template here that is used is in this case will be DNA and the product form. Okay. The template here will be RNA, and the product that is formed is DNA. Okay. So RNA dependent DNA polymerase will use RNA as the template, and it will make DNA. Okay. So now this we earlier talked about is the function of reverse transcription. Okay. The conversion of DNA to RNA is transcription, but the conversion of RNA to DNA is basically reverse transcription. So this is found in certain retroviruses, and in labs it is used to make complementary DNA from cDNA, which can later be utilized in RT-PCR. Okay. So this is same thing is given in this figure. And the fourth term was DNA dependent DNA polymerase, which is where the template used will also be DNA and the product formed, which will be also a DNA. So this is the normal DNA. This is used for replication or for repair that we were talking about. That even for forming a new strand or replication of DNA or for DNA repair, this polymerase is used, which is DNA dependent DNA polymerase. Okay. So this is a small cheat code for you to remember the functions of the four polymerases, that is RNA dependent RNA, DNA dependent RNA, RNA dependent DNA, and DNA dependent DNA polymerases, of what their templates will be, what their product will be, and example of these enzymes. Okay, you can use this to remember. So now coming back to the question, the correct answer to this question will be RNA dependent DNA polymerase, because that in reverse transcription, RNA will be used as template and it will synthesize DNA from it. So the template will be RNA and the product will be DNA. This is the reason it is called as RNA dependent DNA polymerase. It is found in retroviruses and in making of cDNA from RNA. Okay. So the answer to this question is C, RNA dependent DNA polymerase. Reverse transcriptase is an example of this. All right. Moving on to our next question. Which of the following is the example of negative selectable marker gene? The options are MAN A, Zyla, Dog R1, and HPT. So we have one answer C. Anyone else? Okay, so let's talk about this question. First, let's understand what a negative selectable marker gene is. So basically, a negative selectable marker gene it will be the one whose presence will make the cell die, or it will fail to grow under particular condition. Okay, so the other type is called as positive selectable marker gene. Okay, so positive selectable marker gene will help uh, the cell to survive under certain conditions where normal cell is not able to get survived. Okay, so for example, if there is certain kind of antibiotic which is present, okay, so that antibiotic will kill the normal cells in which the resistant gene is not present. But suppose there is a cell in which there is a gene which is resistant to that particular antibiotic. So only this cell will grow on that media, but the one which does not have this gene will not be able to grow on the media containing antibiotic. So this is the type of positive selectable marker. But for negative selectable marker, the presence of that gene in the cell will cause its death. Okay, the cell will die because that gene is present and it is getting expressed. This is called as negative selection. Now, the common examples is HSV-TK, which is also called as herpes simplex virus, thymidine kinase. So, basically, the cells which are carrying this will become sensitive to compounds like janky clovir. Okay, this is a kind of compound which will, in normal cases, not kill the cell. But if there is a negative selectable marker like HSV-TK, the cell will get killed in the presence of this compound. Second one is CORD-A, which is also called as cytosine deaminase. So it will basically convert fluorocytosine into a toxic compound called fluorouracil. And this compound, because this compound gets accumulated inside the cell when the gene is expressed, the cell will also die in this case. Third example is Barnase. Barnase is a ribonuclease which can kill the plant cell when this particular gene is expressed. Okay. So now the four genes name which was written in the options, let's understand their function. Man A is a positive selectable marker and it will help the transform cells to use mannose from the media. Xyla is basically a positive selectable marker in some systems which will help the plant use xylose or xylulose, which is related to metabolism from the media. HPT is a positive selectable marker which gives resistance against hygromycin. Hygromycin is an antibiotic which is used in mostly plant selection medias. And the cells which are expressing HPT gene will be able to grow in the presence of hygromycin. Dog R1 or DOG R1 is a type of negative selection or the counter selection. So, the correct answer to this question will be option. Okay, this is wrong. Option D is not correct. DOG R1 will be the correct answer because that is an example of uh, negative selectable marker. Okay, so it should would not be D, it should be option C. Okay, so I hope this question was clear. Let's move to next question. Choose the correct option regarding the scoreable marker. Scoreable markers are used to identify genetically modified organisms based on visible traits. Scoreable markers do not require any specialized detection methods. Scoreable markers are also antibiotic resistant genes, and scoreable markers will help in distinguishing transform cells based on an observable character. Now, remember, this is an MSQ question. Okay. I hope all of you understand when the question is MCQ and MSQ because it is not mentioned. If these check boxes are square, then in that case, it will be an MSQ question. And if they are circle, then it will be an MCQ question. In case someone doesn't know. So this is an MSQ question. Okay, so let's talk about this question. First, let's understand the meaning of scoreable markers. So, scoreable markers basically are also called as sometimes quantifiable markers. And these marker genes will be used to identify the transformed cells or organisms by some observable character. Okay, so this observable characters, like when we talked about genes like NPT or HPT. Okay, so these are basically survival dependent. Okay, the cells will either survive in the presence of this when the gene is expressed, or the cell will die. Based on this, it is called as either positive selection marker or negative selectable marker. Okay, so this was the concept of selectable markers. But scoreable markers will not depend on the survival of the cell. Instead, there will be some other observable character which will help us to identify whether the cell or the organism has been transformed, or the gene that we require getting expressed in that cell or organism or not. Okay, so uh, in this case, it is like the simply the concept that you can see the cells getting transformed. Okay, so the best example of this is the best. I mean, the observable characters that you can see, which will be present in scoreable markers, or it can be detectable. Will be either there will be some color change in that organism, or there will be some kind of fluorescence produced, or some light will be produced, or some other visible trait will be produced, which will help us to visibly identify that the cell that is being produced now is transformed, and the gene that we require is getting expressed in that cell. Okay, so this is simply based on observation. So these can be some characters on which observable characters can be chosen. Now, there are some very common examples of scoreable markers or genes, or some traits that can be observed, like use of Gus gene. So, Gus gene will basically give blue color after staining. If you see the image on this side, this is an outcome of expression of Gus gene. This blue colored plant that you can observe, this is because the Gus gene is getting expressed, and once the gene gets expressed, the staining, so basically in this I think X-gal is used as a substrate, and after staining with that, the blue colored organism, or if it is a bacterial cell, then blue colored colonies are produced. Another common example is use of GFP, which is called as green fluorescent protein. So it will give basically green fluorescent color under UV light or blue light. Okay. Third one is luciferase enzyme. Luciferase gene, when gets expressed, it will produce light and it can be visible in the organism. There's one more example which is called as YFP, or yellow fluorescent protein. Again, it will produce fluorescence but of yellow color. Now, why they are called as scoreable markers? Because you can easily visually tell whether the cells are getting transformed or not, and it can be scored, or it is a quantifiable character as compared to whether the cells are dying or surviving. I told you about the difference from selectable markers, that scoreable markers will identify the transformants by appearance, as compared to the selectable markers, which will select the transformants based on survival on the selective media. This is the exact examples that I used. HPT and NPT2 are the selectable markers. HPT is for hygromycin and NPT is for kanamycin. And GUS and GFP are the example of scoreable markers. Okay. Now coming back to our question, the option A, scoreable markers are used to identify genetically modified organism based on visible traits, is correct. And option D, scoreable markers help in distinguishing transform cells based on observable characters. Okay. Okay. So why the other two options were wrong? Because the third statement was scoreable markers do not. Second statement was scoreable markers do not require any specialized detection method. So this is wrong because for use of like For identifying the protein like GFP or YFP or luciferase also, there has to be some technique like some UV light or blue light or some special microscopic techniques utilized to observe these characters. Sometimes they are not visible by naked eyes. So B option is incorrect. C was scorable markers are always antibiotic resistant. No, it has not. Those are basically selectable markers. These genes are not antibiotic resistant. So option A and option D are correct in this case about scorable markers. Okay, next question. What is the main advantage of using molecular markers in backcross breeding? The options are it accelerates the selection of desirable traits with higher accuracy. It eliminates the need for a donor plant in breeding programs. It allows direct modification of genes without crossing, and it replaces the need for conventional breeding techniques entirely. Okay, so let's talk about this question. The main advantage of using molecular markers. Okay, so by now we have talked about a lot of molecular markers in previous session as well, but let's just recall it. So molecular markers are basically identifiable DNA sequence variations in the genome that will be used as landmarks to track either genes, traits, or genomic regions. Now, in simple definition, basically they are small flags on our DNA, okay, that will help identify the one genotype from the other genotype. Okay, their presence might be varied, their numbers might be varied, or their locations might be varied, based on which we can determine whether the two genotypes are same or not. Now, why they are helpful? Because they are helpful for breeders and for researchers to identify whether the plant is carrying a desired gene, to study the genetic diversity, to construct linkage maps, assist in marker assisted selection, and track introgation in backcross breeding. So the important features of molecular markers are that they are not affected by the environment. They are detectable at the DNA level and often independent of plant age or tissue type. Now, these are some examples of some common molecular markers: RFLP, RFAD, AFLP, SSR, ISSR, and SMP. We have talked about all these markers in our previous sessions. So if you want, you can go and check that video on YouTube. So the correct answer to our question is that molecular markers will accelerate the selection of desirable traits with higher accuracy in case of backcross selection. Okay, so how this will happen? So in case of backcross breeding, molecular markers will basically target certain target gene or the QTLs, and also in the plants with more recurrent parental genome. And this will make the breeding process more faster, more accurate, and less dependent on phenotype. And this especially is useful when the traits are hard to measure or they are expressed later in later stages of the plants, and whether they are influenced by the environment. Okay, so option A was correct. Now, why others are not correct? B was because donor parent is still needed to provide the desired gene, so it cannot be replaced. Now, molecular markers will, C was molecular markers will not directly modify the genes, but they will only help in selection. And for D option, the support the support is basically for conventional breeding and not to replace it entirely. Okay, so D option was if it replaces the need for conventional breeding. No, that is wrong. C was it allows direct modification without crossing. No, molecular markers cannot basically cause direct modifications. And B, it eliminates the need for donor parents. No, in case of backcross breeding, donor parent is an absolute necessity, so it is also wrong. So option A is correct, that it accelerates the selection of desirable traits with higher accuracy in case of backcross breeding. Okay. So our next question is, which of the following statements correctly describes the role of co-integrate vector in agrobacterium mediated transformation? The options are: a co-integrate vector is formed by recombination between a helper plasmid and disarmed DI plasmid, facilitating gene transfer. Co-integrate vectors do not require homologous recombination for integration in agrobacterium genome. Unlike binary vectors, co-integrate vectors completely lack tDNA sequences, and co-integrate vectors function independently of agrobacterium and directly transfer genes into plant cells. Coming back to this question. So, in case of agrobacterium mediated transformation, there is a co-integrative vector system that will work by the concept of homologous recombination. Okay. So, in case of homologous recombination, there will be two vectors, okay, that will be used. One will be our donor vector and the other one will be a recipient vector or recipient molecule, okay. And a disarmed Ti plasmid will be present in the agrobacterium. Now, in case of now if you look at this figure, how this co-integration basically works is that in donor vector and in the recipient vector, there will be small nicks which will be produced on the single strands of the double stranded uh, 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 basically molecule. Okay. Now ligation will happen where there can be two steps. One can be that DNA synthesis will happen at the four places. Okay. So basically these strands will combine. These strands will combine and co-integrate, and this will lead to homologous recombination by forming replicative transposition. So in this case, the transposon which is present in the donor molecule will get transferred to the recipient molecule and it will get attached here in replicative transposition. But there can be another case where there are additional cuts after ligation, and this entire fragment or the entire transposon is transferred only to the recipient molecule. It will be deleted from the donor molecule and get transferred to the recipient molecule. This is the concept of co-integrative plasmid, and it will contain tDNA borders. It will contain our gene of interest, and it will also contain Vir genes, also called as virulence genes, which are needed for the transfer to the plant cell. Okay, so now based on this, the correct answer will be that a co-integrative vector is formed by recombination between helper plasmid and a disarmed Ti plasmid, facilitating the gene transfer. Why the other ones are wrong? Because second option was co-integrative vectors do not require homologous recombination. No, it requires homologous combination, so that is why it is wrong. C was unlike binary vectors, they lack tDNA. No, tDNA sequence is present in the co-integrative vector, so this one is also wrong. D was co-integrative vectors function independently of agrobacterium and directly transfer genes into plant cell. No. Co-integrative vector system requires agrobacterium for the transfer of the target genes in the plant cell. So option A is correct, that co-integrative vector is formed by recombination between a helper plasmid and a disarmed Ti plasmid for facilitation of gene transfer. Okay, I hope this was clear. So our next question is, which enzyme is primarily responsible for breaking hydrogen bonds during DNA replication? The options are DNA polymerase, helicase, ligase, and primase. This is an easy question. Okay. So talking about this question, the four terms that were given in the options, let's understand those first. So first was DNA polymerase. We have already talked a lot about DNA polymerase, that this enzyme will basically synthesize new DNA strands by adding nucleotides to the 3' end of a primer. Okay. So it will build a new daughter strand and it will work in 5' to 3' direction. And like we talked about, that DNA polymerase 1 also has the proofreading activity. B was helicase enzyme. So the function of helicase enzyme is to unwind the double-stranded DNA. Okay, so it is wound in this structure, and to unwind it for the replication to happen, this particular enzyme, which is helicase, it will break these hydrogen bonds. Okay, between the two complementary DNA, it will break that and separate the two strands so that the polymerase enzyme can fit in and it can perform its function. This structure is called as replication fork. Okay. So this the two parental strands one gets separated and open into forming a replication fork, and this is how the DNA strand gets unwound. Okay. Now our C option was ligase. Okay. So ligase, the function of ligase is that it will join the broken sugar phosphate backbone of the DNA. So suppose if we cut with some restriction enzyme and the two sticky ends are produced, the function is to join these complementary sequences by hydrogen bonds. Okay, this step is called as ligation, and this is done by the ligase enzyme. In natural processes, it can also seal the Okazaki fragments on the lagging strand, and it this is done by forming the phosphodiester bond. So ligase does not open, but it joins the pieces together. And the fourth one was that the primase enzyme. So primase enzyme basically synthesizes short RNA primers. Now these are used for providing free 3 dash OHN for DNA polymerase to start synthesis on the opposite strand, and like multiple on the opposite strand and one in case of leading strand, because DNA cannot start the procedure of replication from the scratch. It needs a primer first, and that primer is synthesized by the enzyme primase. Okay. So now coming back to our question, the correct answer to the question was helicase enzyme, because helicase enzyme unwinds the DNA by breaking the hydrogen bonds between complementary bases, and this will separate the two strands, forming the replication fork. Okay, so by now you understood that why other options are incorrect, because uh, option B is correct. DNA polymerase is utilized for the formation of new strand, new daughter strand, so it is wrong. C was ligase, it joins the fragments and not break it, and primase is used for synthesizing the primer for the initial step of the replication. Okay, so moving on to our next question. What is the purpose of using selective markers in genetic transformation? The options are to enhance plant growth, to identify transform cells, to replicate inserted genes, and to increase transformation efficiency. Okay. So let's talk about this question. Okay. This was very easy. We have been talking about it all along. The correct answer is to identify transform cells because selectable markers are genes which are introduced in the cells along with the gene of interest so that only successful transform cells will grow under selective conditions. Either they will grow or survive under specific conditions. We talked about the example as well, NPT2 and HPT. NPT is kanamycin resistance and HPT is hygromycin resistance. So when the cells will grow on the media which contain these antibiotics, only those cells which are expressing the selectable marker genes will be able to grow. Okay. The other answers options. So option B is correct to identify transform cells. Option A was to enhance plant growth. No, selective markers do not enhance plant growth. To replicate inserted gene, no, the organism does that by itself. And D is to increase transformation efficiency. No, these selective markers have nothing to do with the transformation efficiency. They are just transformed along with the gene of interest. Okay. So the last question is agrobacterium tumefaciens is widely used to create recombinant plants. Which of the following gene helps the bacterium to sustain itself in the rhizospheric region of the soil? The options are Vir genes, opine catabolism gene, auxin synthesis gene, and cytokinin synthesis gene. Okay, so let's understand this question. Um, so basically, Agrobacterium tumefaciens transferring with transferring the gene of interest in plants using this gene requires a, a TI plasmid. Okay, a helper TI plasmid is required for this process to happen, and that helper TI plasmid will contain multiple genes which will facilitate this particular process. Okay, and that genes will also help in the survival. Okay, so now how this exactly happens? So some of the most common genes that are present is Vir gene, which will help in tDNA transfer. Okay, Vir genes are also called as the virulence genes, and there are multiple Vir genes which is present in the TI plasmids, and each will have their different function while transforming. Okay, the second one is the set of plant hormone genes like auxin and cytokinin synthesis genes, which are present within the tDNA sequence, and it will help induce the formation of tumor in the plant. Okay, and the third one is the use of opine synthesis genes. Okay, so the transformed plant cells will produce opines, and these are the unusual compounds which is used by the bacterium for their carbon and nitrogen sources. So normal cells do not produce these opines, but once there is a tumor formed inside the cell, the transformed cells will start producing opines, and then these are basically used by the bacteria for its growth as a source of carbon and nitrogen. Okay, so basically, if you're talking about the survival, every organism requires the source of carbon and nitrogen for their survival. So, in case of agrobacterium, the opine catabolism genes will help the agrobacterium to utilize the opines as nutrients, and that will support its survival around the infected root region. Okay, so this is what our question was. This is the structure of a normal TI plasmid. Okay, so these are various Vir genes. Uh, okay, so these were various Vir genes in our structure, like Vir G, Vir C, Vir D, and Vir E. These genes will help in transfer of the gene. Okay, then there is Vir B and Vir A. So all these have different functions. You can see like Vir E will help in tDNA movement. Vir D will help in tDNA processing, where C will help in tDNA processing and recruitment, where G is a virulence gene activator, where B is for translocation, and where A is a sensor kinase. Then comes the TI plasmid replication side, TI plasmid conjugation side. Opine catabolism is done for opine uptake and degradation. TRA is TI plasmid processing gene, and this is the left border and the right border sequence in which auxin, cytokinin, and opine synthesis genes are basically produced, and the genes in this part basically gets transformed in the cell and gets integrated in the plant genome. Okay, from left border to right border. Okay, it will get integrated in the plant genome. So now, according to this, our correct answer will be the opine catabolism genes will help the agrobacterium to utilize opines, and this will help in the survival. Why other? So the correct option is B, which is opine catabolism gene. Why others were wrong? Where gene is basically involved in gene transfer, and it has nothing to do with the nourishment. Auxin synthesis genes is a plant hormone, and it will help in the tumor formation in the root and not in survival of the bacteria. And C was cytokine synthesis gene, again a plant hormone, and again it is helpful in tumor induction or tumor formation. So the correct option is B, opine catabolism gene, which will be which will help the bacteria to sustain itself in the rhizospheric region or the root region of the soil. Okay. All right. I hope these questions were helpful for you. If there is any doubt, you can ask. Or otherwise, we will end this session and we will meet again next week, Tuesday, same time.